Hi, Mrs. Murdock, and this is a review session, and what we're going to talk about in particular here is how an action potential moves along a neuron. So an action potential has a lot of moving parts to it, but to anchor ourselves, what I'm going to start by doing is I'm going to draw a simple nerve cell. So let's say we have a nerve cell, and it's going to be really oversimplified, and I'm not the greatest artist. And I'm going to draw the axon abnormally thick. This is not a typical axon. And an axon can be as long as your arm. It can be as long as from your spine down to your toes. So kind of picture that. And then we have the end of the axon here. Okay. So over here you have the cell body. So just like any cell, you have, in here you have cytoplasm, and you have a nucleus, and you have a cell membrane. Remember, this is an animal cell, so there's no cell wall. And what happens along the cell membrane in a neuron, this whole thing is a neuron. Write that so that you'll know I'm talking about a neuron, which is a type of cell. Right? This is all about diffusion. Diffusion, passive and active transport actually are happening in an action potential's journey from this part of the neuron all the way down to the end. So first of all, let's talk about something called resting potential. So resting potential means that along the outside of, just outside the membrane, the cell membrane of the neuron, there is a slight positive charge. And I'll put it on both sides because resting potential is maintained all the way down. And on the inside, in the cytoplasmic side, it's negative. It's slightly negative. And the things really responsible for this difference, for this gradient across the membrane. Remember, gradient just means there's a difference in concentration of things from one side to the other. The things that are causing that gradient are two ions called sodium and potassium. Okay. And in resting potential, during resting potential, which this is picturing right here, and which really goes all the way down. I'm just not going to draw all of that. Resting potential has to be maintained. It actually takes work to create a gradient across a membrane. And you may remember in your travels through whichever chapter talked about osmoregulation, that there are membrane proteins all through here, right, that allow things to travel through membranes. And in a neuron, there's one particularly important membrane transport protein that kind of looked, if I draw a big thing of it, kind of looked like this. It looked like this on your mastery check, right? And it has the ability to push out 3 sodiums for every 2 potassiums that it brings in. 2 potassiums come in, 3 sodiums go out. And this is called the sodium-potassium pump. Anything that is a pump, any transport protein with the word pump associated with it tends to be an active transport protein. It needs ATP in order to do this particular job. And the fact that it's bringing out, like if I put it up here, here's a pump right here, that it's pushing out more sodiums, right, than it is pushing in potassiums. And I remember when I was learning this when I was 17, 18 in this same class, I was confused by, but wait, why isn't it bringing in chlorine instead? Since chlorine is negative, doesn't it make sense to have why isn't it pulling in a negative ion and pushing out a positive ion? Wouldn't that be easier? Well, we don't know why the system evolved the way it did. But in pushing out more sodiums than it brings in potassiums, that makes for slightly more positivity on the outside than positivity on the inside. And that does create the resting potential. I always thought resting potential was kind of a misnomer, because in order to maintain resting potential, the cell has to expend energy. It takes energy to kind of pull back on that spring. That's exactly what the nerve cell is doing. It's preparing for the system to be let go during the action potential, right? So the resting potential is what the neuron is sitting in as it waits for a stimulus to reach this side. It's waiting for a stimulus here. Some kind of stimulus to start the action potential and open up sodium channels. That's the second type of important transport protein in this story. But it needs a stimulus to do it. And as soon as the stimulus is strong enough, what happens is that now you're going to open sodium. I'll make it a different color. Make sodium green. How about that? There. So if you open up sodium channels that weren't open before because this got stimulated, now all of the sodium will passively flow back through, right? So the action potential begins here. And as the sodium flows back in, what happens? It reverses the action potential. So now you have more positive on the inside and more negative on the outside, which is what an action potential is. An action potential is the depolarization of the membrane, and it moves this way, reversing the pluses and minuses all the way down the axon. It keeps going and it keeps going until it reaches the end of the axon terminus, and then it might jump across to another neuron or a muscle cell that it might be telling to do something, or maybe to something in another neuron in the spine. It takes the nerve impulse to another part of the body that will deal with it, right? But the only part of the story I really want you to worry about is what's going on in this axon. So we've got this axon potential that is now traveling, right? And we reversed all of this resting potential. We reversed it. And here it comes, and now it's positive inside the cytoplasm and negative just outside the cell membrane. Well, that can't stay that way. The cell can't just stay that way. I mean, eventually it's going to get over here and the impulse will travel over there, and that the neuron will be done. There has to be a way to put things back to rights. So that's the job of the potassium pump. I'll make it a different color. Let's make that orange. Okay. All right, so behind the action potential, now that the sodiums have all built up inside, now we're going to have the potassium have the chance to reverse the polarity. So now the potassiums are allowed to flow and all the sodium gates are shutting. So the potassiums flow and the potassiums flow. And this is repolarizing the membrane. Putting it back to, now we're pluses on the outside, negatives on the inside, because the potassiums are building up out here, right? And that's repolarization of the membrane, repolarization. And it keeps going until you almost get hyperpolarization. It almost gets too positive on the outside, right? So the action potential goes away. We've got the charges on the right side now, right? We've got it back to what looks like resting potential, but the potassiums and sodiums are on the wrong sides. So finally what happens is it goes back to our good old sodium-potassium pump again and says, okay, we need you back because everything's all messed up. We have got to put it back the way it needs to be. Because we can't have the potassiums and sodiums on the wrong side, because when we need another action potential, if there's another stimulus, the passive gateways only work in one direction. So the last thing that happens is that the sodium-potassium pump is going to go, all right, let's get, because you have potassiums here, right? So the potassiums are going to be pulled back in, and the sodiums, which all were going in here, are going to be pulled back out. And I'm sorry that looks a bit messy, but I think if you look at one of the videos that's in one of the ed puzzles, it shows it a little bit more clearly. But in any case, on the graph, and I know you guys were angsting a bit about that graph, right? The graph is measuring what's going on inside the cytoplasm. So in the cytoplasm, it's either going to be negative or positively charged. The resting potential is, I think, right at 70, negative 70. I don't know, I don't think I have in my mind what it actually is. But it goes up to positive 30 during the action potential. So it stays, the resting potential is like right here. So that part is where you have, you do have salty bananas. So you have negative inside, resting potential, negative inside, positive outside. And if you could put a little electrode in there, it literally tells you what, I think it's like, can't remember, it's like negative 50 or something like that. And if you stimulate it enough, it goes above resting potential. It has to hit this threshold here before it will fire. And then that's when the passive sodium gates open, right? So sodium flows here. It hits a maximum, and then the sodium gates fall, and then the potassium flows on the way back down to put it back to rights. And it hyperpolarizes a little bit and then comes back up to resting potential here. So this is resting potential. This is depolarization. I miss my old writing pad. Very nice. This is depolarization, right? Leaving resting potential, action potential, right? This is repolarization on this side, putting back things so that the charges are back where they need to be. Right, going back down this way. So we go up this way, sodiums flow out, back this way, potassium flows out, and then it hyperpolarizes just a little bit before it reaches resting potential again. Okay, so once again, the sodium flow and the potassium flow are passive. The only reason they can happen is because the pump, which uses energy, set it up like pulling back on a spring so that there was a gradient, and nature doesn't like a gradient. So when you've got a gradient and a sodium passive doorway opens, then those sodiums are going to flow and create the action potential. So hopefully that is a little bit more clear and a little bit more helpful for you. And I will put that up so that everybody can watch it. But I also do recommend watching Mr. Anderson's version and watching the small animation, which is really clear too. And I hope that was helpful.